So the pandemic has created opportunities for researchers in the areas of testing, surveillance, and treatments. It also spurred technological innovations. For our first session, we're going to be hearing from 3 researchers, starting with Dr. Euan Ashley, who is a professor of medicine and genetics and associate dean of precision health at Stanford Medicine. He's a cardiologist and also directs the Stanford Clinical Genome Program and the Center for Inherited Cardiovascular Disease. Next, we'll have Dr. Idan Yelin, who is the head of the Pathogen Genomics Research at the Kishony lab in Technion, Israel Institute of Technology. He received his Ph.D. in microbiology at the Hebrew University of Jerusalem. And finally, we have Dr. Andra Blomkalns, who is the Rutledge Family Professor and inaugural chair of the Department of Emergency Medicine at Stanford University School of Medicine. She is a national leader in emergency medicine and a strong advocate for innovation in patient-centered clinical practice. So let's start with Dr. Ashley to talk about genomics and innovations in testing. Dr. Ashley, please feel free to share your slides. And as a reminder to the audience, please feel free to submit your questions in the chat bubble icon located in the upper right-hand part of your screen. Thank you.
Great. Well, thank you, Sam, and thank you to all the organizers for the opportunity to speak today. It's really a privilege to speak to this incredible collaboration. You'll see a lot of letters on your screen, I hope, if everything's working well. This is, in fact, the genome of SARS-CoV-2. It's 30,000 letters of RNA. This is actually the protein code which fits on one screen. That managed to stop the world in its tracks, as you know well. This was a quote from Peter Medawar, who famously described a virus in 1977 as a piece of bad news wrapped up in protein. Pretty good description for the current virus attacking our world. My work at Stanford is in genetics and cardiology. My main interest is in cardiovascular disease. We have over the years developed many tools to help understand the human genome in relation to human disease. So it was new for us when all of the labs shut down and we realized that we could perhaps turn our attention and turn some of our expertise towards sequencing not just the humans who are infected with COVID-19, but also the virus. So with a very large group at Stanford and at the Chan Zuckerberg Biohub here in the Bay Area, we began working together to try to understand at a genomic level both the virus, the host genetic factors, which would include both the predisposition factors and also the transcriptome. And so to date, we've sequenced 1,200 patients in the first wave of patients at Stanford, 400 negative controls. And we have been sharing this data since the earliest moment and contributing it to the various collaborations around the world. We have developed scores like this, that is a digital severity score automatically drawn from EPIC, where we can pull the WHO severity score for COVID-19 directly, also on a daily basis. So we can follow patients as they are recruited into the biobank and recruited through the emergency department and through the other parts of the testing organization at Stanford. And we can pull this into the open source portal that's available to the world. This is it here. It's at covidomics.org. There's a preprint just describing our basic method, which is to take the discard nucleotides from the nasal swab and essentially pull both DNA for low-pass sequencing, the equivalent of genotyping, as well as host transcriptome and virus, as well as the host HLA. And here's some of the early interpretation of the data. In fact, the first graph shows you the case count ethnicity by self-report from LA County in 50 to 64 age group. And you can see what we've found since the beginning of this pandemic, that those who are underserved and underrepresented in our society are overrepresented in those patients who suffer from COVID-19. In particular, you can see here the Native Hawaiian and Pacific Islander group in the Bay Area, as well as Hispanics and Blacks overrepresented. The bottom graph actually shows this on a per segment genotype level, where the admixture of individuals is shown vertically. And you can see again the overrepresentation of the Indigenous American Pacific Islander population. Statistically, this is also falls out. These are PCA plots, principal component analysis plots, where it's an unsupervised method of looking at the genotyping of the patients who came in and comparing it to the controls. And the main message is shown here in the text, that there's overrepresentation in cases of East Asian and admixed being predominantly African American and the Native American individuals. This is the very large study that we've contributed data to. It's 100,000 controls, 10,000 hospitalized patients, and it's a genome-wide association study. This is one of these Manhattan graphs where the genome is shown by chromosome on the x-axis and the p-value is shown on the y-axis, and you could tell from the log -10 that we now have significant genome-wide hits shown here. Wherever this red line is surpassed by these dots are significant genome-wide hits. And the reason this is interesting is because, as we all know, there's a very wide, across the age groups, a very wide diversity of presentation, with some patients being completely asymptomatic, but other patients, of course, laboring in the ICU for many days and even dying of this condition. So partly this genome-wide association study is our first attempt to find representative factors that would explain that. And you can see even if the exact genes aren't ones that you recognize, certainly these are autoimmune genes. There are potentially other genes involved in the interferon pathway. The ABO, which many of you may have heard as a predisposing factor to the blood type, is borderline significant here, and we can certainly discuss that if there's some interest. It is, I think, an important criterion for virus entry, but less so for the severity of the illness. And so one more thing just to mention before we move on, we've also been developing a testing system. And again, the emergency department has been very helpful in helping us push this forward. We realized that while there are many tests available, the clinical tests take at a minimum 24 hours. At the beginning, they were costing somewhere between $50 and $100. And we were very interested to see if we could take that same technology, in fact, a PCR-less technology called isothermal amplification, invented in Japan a few years ago, and basically build an at-home clinical grade kit for $1. And we've been working with Manu Prakash, a Stanford professor, who focuses on frugal innovation, the ability to invent mechanisms of carrying out high-level medical diagnostics in this case, but at very low cost and in settings that do not require electricity. And so we're moving forward, in fact, with this pilot. This is the how-to instructions. Just in the next week we'll start, and over the course of next month we'll do 10,000 individuals around the Bay Area. This is a test that if you can boil a cup of water, you can do at home with saliva in around 30 minutes. And so our hope is this could help lead to opening schools and businesses. And as the vaccines that we are keeping our fingers crossed come forward over the next 6 months, it should hopefully be a large part of helping us open these schools and businesses. And with that, I will pass on to the rest of the group. Sorry about the technical issue with the slides.
Thank you, Dr. Ashley. That's very interesting to see, COVID at the sequence and at the genomic level, and I'm also very excited to partner with you on the saliva test for the emergency department. Next, we have Dr. Yelin. Please feel free to share your slides.
Okay, so thank you again, and I'd like to take this opportunity to present some work that we've done during the last couple of months. We've switched from microbiology and antibiotic resistance, another pressing issue, into COVID-19, and specifically COVID-19 testing. And I'll show you two parts of the work that we've been doing. So during this epidemic, it became very clear that testing, and specifically RT-qPCR based testing, is crucial, and it's important both for diagnostic and epidemiological purposes. But however, testing capacity worldwide is limiting, and we and others have suggested that sample pooling can save resources and increase throughput. So the basic idea, very briefly, is that if you test a large population, you usually need one test per person. The problem is that when prevalence is low, let's say in this group of 24 people, only one test is going to turn out positive, identifying the patient, diagnosing just one patient. So the idea of pooling is that you divide the population into a few groups. Each group is tested in a single tube, and while most tubes are going to end up negative, when you have one tube that is positive, you can go back to the initial sample, check each one of these samples, and find again the single positive patient. That's the basic idea. We didn't invent it, obviously. And then again, let's say the improvement or the efficiency depends a lot on the prevalence in the population. And while in extremely low prevalence, like 0.1%, which is obviously fictional at this point of the pandemic, you can save way over 90% of resources. So even in a higher prevalent, let's say between 5 and 10%, at least half of the resources can be, you can kind of decrease the use of over 50% of the resources, which is still quite significant. So quite early on, actually we did it already in March, we've shown that COVID-19 tests are can be diluted, and if you take a positive sample and dilute it in many negative samples, you can still see a very clear signal here. You can see an example where a single positive sample is diluted with up to 63 negative samples, and right, you see it in a higher CT, but you still clearly see the sample. And you can actually also do this pooling prior to RNA extraction, which is probably more realistic for diagnostic labs. And if you do something, again, very realistic, of pulling 8 samples together prior to RNA extraction, you have an increase in CT of just about 2.9, which keeps most of the samples, if not all of them, detectable. But an issue here that obviously arises is the issue of sensitivity, because we're really playing with the sensitivity of the test. We are somewhat decreasing the sensitivity of the tests. And this brings me to the other project that we've very recently concluded, which is trying to identify the very basic false positive rate of COVID-19 tests. So these tests are notoriously known to have a high false negative rate, and some studies have reported rates of about 30%. And one can argue that a false negative result is actually more harmful than a false positive, that these tests actually don't suffer a lot from. Because a false negative result can impede the ongoing efforts to break the chain of infection, right? So, but what is really the false negative rate of COVID-19 test? It's kind of hard to tell. We've chosen an approach that looks retrospectively at data. And the approach says something like that. Let's say a person, and it happens a lot, that a person is tested, not experimentally, just goes to the physician or healthcare provider and is being tested a couple of times longitudinally over many days. And some of the tests are going to be positive and some negative. Then you can retrospectively look at it and assign a sample false negative state. Based on what? Let's say you have a test that came after diagnosis with COVID-19, but is followed by another test which is positive. Then this test is almost certainly a false negative result. This is not a reinfection within 2 days. I think we can all agree on that. So we took a lot of these series and assigned, okay, this test is true positive, this test is retrospectively can be assigned as false negative, and we look at the data. So we collaborated with Maccabi Healthcare Services, which is a large healthcare maintenance organization here in Israel, the second largest in Israel, with over 2 million patients. And that's a data set of more than 7,500 patients who had longitudinal data. They had quite a few tests over time. And in this data set, we could identify almost 2,000 false negative tests, and you can see the numbers here, 6,700 for true positive tests, which gives roughly a false negative rate of 23%. Okay, so it gives a number, quite a high one, I must say. But then we thought, maybe now we can ask other questions. Let's say, how do these tests distribute how are they distributed over time? And we had some data about the patients, such as sex and age. Can these be correlated with false negative results? And the answers are obviously yes, yes, and yes. So we found that false negative rate actually increases over time. And when we say over time, we mean time since diagnosis. And you can see that while in the very early days, let's say 5 or 6 days after diagnosis, the false negative rate is about 10%. It then gradually increases and ends up being around 40% later on. And something also happens with age, especially during the first few days, where patients who are younger patients, let's say in this case below 40, have a higher propensity or a higher risk of a false negative result. And the same was found actually for sex. False negative rate is actually smaller for women. Okay. And all these results suggested, okay, what can underlie these associations? What can be the result for these differences in false negative rate? And we thought maybe the viral load itself changes along, again, time, age, and sex. And indeed, if you look here, you can see the viral load, again, days after diagnosis for patients over 40 or below 40. And you can see that for patients below 40, the CT value is actually higher. For that, we obtained a large data set kind of directly from the PCR machines in Maccabi Healthcare Services. And you can see that again, especially in the early days, viral load for younger people tends to be lower. The CT is actually higher. So this strongly suggests that differences in viral load affect false negative rate for these patients. And with that, I'd like to conclude and thank the wonderful team. I'm part of the Kishony lab at the Technion. You can see all the people from the Technion involved in this study, as well as people from the Rambam Healthcare Campus were involved, especially in the pooling experiments very early on, and Maccabi Healthcare Services for the false negative rate study. Thank you very much.
Thank you, Dr. Yelin. Next, we have Dr. Blomkalns. Please feel free to share your slides.
Good morning and good evening, everyone. Thank you so much for joining. My name is Andra Blomkalns. I'm the chair of emergency medicine here at Stanford, and it's truly an honor and delight to tell you a little bit about the disaster level research opportunities and challenges that were met, particularly early on in the pandemic in the United States. We have many, many stories to tell, and as you can see, the research has exploded around the world, around the country and around the world. This is data actually from today. So lest we think that we're out of this pandemic or out of this disaster situation, we may be ending or going into the worst part of it. Currently, globally, we have 51 million cases, where the US tops the charts with over 10 million cases. Fortunately, Israel as of today was 320,917. Much lower than that, but nonetheless, we see globally that the daily number of cases are increasing. And what that tells all of us, I think, is that research in this situation is so important to transcend countries and continents and cultural divides and otherwise, because the health and well-being of our worldwide population is dependent upon these very, very important collaborations. I'm an emergency physician, so I'll describe what it felt like on the ground in terms of trying to upstand some of these research efforts, because research opportunities suddenly blossomed. They blossomed particularly in what I would say collaboration, and in collaboration both across countries and cultures, but also specific fields. So as we've heard from our previous presenters, very quickly, researchers across disciplines were collaborating in different ways. Within a couple days in early February, I had met Bali Pulendran and Bonnie Maldonado, two individuals I'd heard of greatly, but had never actually met in any situation. We were suddenly at the table trying to think of what do we need right now to do. And this was still when we didn't understand the virus very much at all. We didn't understand what PPE was necessary. There was not widespread testing. The testing was limited to the counties with a very limited number of tests. Stanford had not yet developed its PCR platform. And I remember gathering my researchers literally around the kitchen table one Sunday. None of us were wearing masks. We didn't know we were supposed to do that at that point, and said, okay, we've got to get samples. And the question was, well, what are we going to do with them? Right? It doesn't matter right now. We have to get samples, and we have to get samples. And these samples from these early patients are going to be so critical in terms of taking care of understanding later patients. So, traditionally, we knew a biobanking effort was absolutely necessary. And several investigators across campus stood one up. We stood one up essentially over the span of a week and began collecting samples from both symptomatic and asymptomatic individuals across the spectrum of severity. So those who were asymptomatic to those who were in the ICU, and serial samples over the course of disease, both blood samples and samples obviously for genetics, and nasal swab samples. Another thing that became very evident early is that testing is going to be very, very, very important. And at that point, none of the clinics or neither the hospital, we were not quite sure where to have COVID patients come back and what sort of isolation procedures should we have. So we put together literally a vinyl tent in the rain in an abandoned parking lot to have patients come back and get anterior nasal samples to compare them to their posterior nasal samples over that time, because we were trying to think of ways, how could we test a large population? Research on personal protective equipment, incredibly important. Where there was a limited amount of personal protective equipment, what do we need to keep us, our patients, providers, and the population safe? The cross-disciplinary collaborations, I think, are perhaps one of the best things, if there can be any best things that came out of this pandemic, is the notion of scientists speaking to one another who may have not even understood that the other existed. And I find that it's so incredibly inspiring on so many levels, that now across the spectrum of disease, detection, treatment, and now with prevention, that we're all working together and working together quickly to move forward with research. Funding became an additional opportunity. I think industry, federal governments, and private individuals reached out very quickly to offer additional funding for what was very, very important. There were some challenges, though, as you might expect, and we all know that it's important to conduct research according to regulations and ethics, but those regulations take time. IRBs take time to fill out, and these consents take time to fill out, and that's very, very important in protecting our patients. But then also, a typical IRB may take 3-4 weeks, maybe even a few months to be approved, and we didn't necessarily had time for that. So our IRB worked with us very closely, and my favorite is that we were able to get an IRB approved in 6 hours over a weekend. Which I've never heard of. And I thought that was just particularly uplifting, because everyone seemed to know that it was so important for us to engage in a true sort of disaster format and have whatever resources we had to move forward. We had some issues with individual fears. Not all investigators and research associates and participants wanted to engage in COVID research. Most of the labs, or nearly all of the labs across campus and across the United States, were closed because schools were closed, everything was closing. But even though we have a lot of people who their occupation is to do research and participate in research and make research happen, suddenly we have a group of people who might not be necessarily willing to engage with active COVID samples or an active situation that may be potentially harmful, and if at the very least, very scary for them to engage with. Equipment challenges. PPE, notably, we were running low on PPE. We fortunately never ran out of PPE, but the notion of you need extra PPE for research. So we don't want to deplete the community stores or the hospital stores. So we were trying very hard to get PPE in small batches from other types of industries and organizations. We got a huge batch from a semiconductor plant because they need PPE when they're manufacturing their semiconductors and on the floor, and they weren't working anymore. So they were able to provide a batch specifically for research. It wasn't a batch large enough for a hospital. It was a large enough batch for our research and our team so that we could get the samples together to be able to help researchers all over campus and all over the world. And at this point, there are almost conflicting priorities. We have several therapeutics that are engaged in EUA, that are in trials. Currently, we fortunately don't have enough patients necessarily to go around. So whereas there is a great need and a great number of patients initially, at this point, which therapeutic trial do you enroll a patient in? Don't know which therapeutic is the best or what might be best for that specific patient. And there are several choices. And that brings up some interesting conversations and potential interesting conflicts. And patients themselves are more savvy now and understand and actually request certain experimental therapeutics, which is problematic for a typical research project. However, I feel as though the rapid advancement of therapeutics, particularly the monoclonal antibody treatments, we have a vaccine on the horizon, and this is within 8 months, essentially, of the pandemic hitting its worst. So I feel like COVID-19 has brought truly the best out of science, worldwide science, and the best out of researchers as we work together to help stem this pandemic. Thank you.
Thank you to all the presenters. We have a few minutes to propose some questions to this panel. I'd like to start with Dr. Ashley. To follow up on your presentation, can you tell us a little bit more about how the genomics or immunology explains the diversity in the presentations of the disease that we're seeing?
Yeah, thanks, Sam. It's a great question. And I think that was really the major motivator for looking at the genetic factors for the host and of course, the virus. We have, as you know, as I noted on the slide, begun to discover some host factors in particular. There are some viral factors as well, but I don't think that we've really got close yet to something that would be useful, like a predisposing score. You might have heard of polygenic risk scores. They can be helpful. We're certainly generating those. I think another factor that is pretty important that I didn't touch on, but that clearly we're seeing great evidence for in the literature is pre-existing antibodies. And there's been some debate around this, but I think the debate is really closed. In particular, many will be aware of the seasonal coronaviruses that many are exposed to. And the question came up as to what extent there might be neutralizing antibodies pre-existing as a result of those that are cross-reactive. And in fact, I think in the last few weeks, I would say we've seen the best evidence yet that those indeed do exist. And they do likely contribute at some level towards the relatively mild illness that in particular our younger patients have been experiencing. I think those cross-reactive antibodies were found most likely to occur in the groups that are at school that would perhaps be most likely to be suffering from those seasonal coronaviruses. So I think in the end, there clearly are genetic factors, and particularly those with very severe illnesses who were previously without risk factors. I think likely they have an underlying genetic predisposition, likely in interferon pathways. But for the larger majority, I suspect that cross-reactive antibodies are a significant component.
Thank you. Very, very interesting. Dr. Yelin, besides, you talked a little bit about how time, gender, and age influences the false negative results. What other factors have you found could be associated with that as well?
Actually, we didn't have a lot of data for these patients. We mainly had the very basic demographics, age, sex, and we took out of the data the date of diagnosis. So that's kind of all that we had, and all of these actually were highly associated with it. I don't know what other things to expect. We did not have yet the data of like underlying conditions, let's say diabetes, etc. We can have this data, and we'll probably look at it later on, but not right now.
Could you comment on viral load in association with false negatives?
Yeah, sure. So, yeah. So I think viral load turned out to be seemed like it's the underlying cause for the false negative rate. It's associated with age, sex, and time since diagnosis just in the same way. So we think this is the factor. But basically, it means that from the patient records, you can put let's say a patient gets a negative result, you can put like a tag of quality of how sure we are that this patient really, that this result is truly false negative. But other than that, we actually did look to see whether at a single patient level, we can see a correlation between a viral load and a false negative result. So we looked at actually the sample that comes after the sample that's, let's say, either true positive or false negative. A positive result that comes after a result that is false negative or true positive. And we saw that for false negative results, the viral load in the following sample were actually lower at a single patient level. So we're quite confident that that's the reason for these results.
Great, thank you. And Dr. Blomkalns, given that emergency medicine is at the kind of front door of oftentimes disasters, what do you see as opportunities for emergency medicine when it comes to research in future disasters, whether infectious or non-infectious?
Sure, thank you so much, Sam. I think, as you said, since emergency medicine is at the front door, we have to recognize the important part that we have in terms of identifying patients with whatever affliction or whatever condition, and the need to do research for that initial period, and how important it is to start collecting samples and enrolling patients from the very beginning. I think we've learned a lot from this particular pandemic on how to mobilize research and get it up and going. Many of researchers, we consider grant opportunities and request for proposals, and they have a deadline, and then suddenly you're met with a situation that has no proposal, has no funding, has no deadline, and you have to just put it together and start moving, even if the end goal is unclear and unknown. So I look forward to trying to figure out how to do that more efficiently and more quickly. I think everyone did a wonderful job in this situation. But for whatever that next disaster is, whether it's infectious disease or not, it'll be important to know how to mobilize those efforts quickly without the traditional mechanisms of what we're familiar with in research.
Can you comment a little bit more about, and you alluded to the collaboration, how you had to work with other researchers that you hadn't worked with before? Could you comment in terms of the role of collaboration, and how would you set that up to be better positioned to work with your colleagues in the future?
Well, I think the conversations early were just absolutely amazing and very, very interesting. And it was the notion of, what do you need, almost speaking to what does everybody need around the table to help their research move forward. And for some people, it was, monocytes. For other people, it was genes. For other people, it was plasma. And other people, it was serum. Other people needed nasal secretions and serial nasal secretions. So the notion of finding out what people needed, and then also employing their help in getting those samples. It was very clear from the beginning, our first United States pandemic crunches were with cruise ships. And it was the notion of, okay, who's willing to go and get dressed up in PPE and go to this cruise ship and start getting samples, and potentially for another researcher. And that's the thing. It wasn't clear where these samples needed to go or what needed to happen, but we knew we needed these samples. So I think conversations are early, and a willingness for everybody to chip in a situation that is otherwise difficult and unknown.